It's a pleasure to be here. I was asked to give everybody a brief update on genomic science and what the impact is for global health. So let me start right in. There we go. So in fact, it was just about 10 years ago that a meeting in the White House with President Clinton, we announced the first draft of the human genome. Since then, things have been changing quite a bit. 3 years ago, we published a paper on the first complete human genome. In that case, it was actually mine, and it helped to understand how different we all are. In fact, much more so than we thought 7 years earlier. And we all differ between 1% and 3% from each other. Now, there's lots of personal genomes being done. The cost of sequencing genomes has gone from $5 billion down to around somewhere between $5,000 and $50,000. It's going to keep going down over the next 4 or 5 years to be under $1,000 is what all the predictions are. So that's going to make it much more widely available. But just with some of the early ones, Vanessa Hayes from my institute recently led a team to do 3 key genomes in Africa, including Desmond Tutu. And the Venn diagram in the middle, in fact, shows within Africa, different populations there. There's more genetic diversity there, in fact, than we find comparing my genome to a Han Chinese to one of the Bushmen in Africa. So we all evolved out of Africa into distinct populations, but the diversity that originated in Africa is much greater than the rest of the diversity. And that's very important in terms of anything to do with health, is understanding that diversity even within Africa. Now, we have 23,000 genes, and as we speed up personal genomics, and you can all get your genome read in the future, we will start to have databases that enable predictive medicine and preventative medicine. But it turns out our 23,000 genes are only a part of what we have. And we use these same tools to sequence the microbes associated with this. And I'm sure you'll think this is totally appropriate for lunch, but each of us have a large repertoire of bacteria. This is called the Human Microbiome. When we're born, we have no bacteria associated with us, but we acquire over 200 trillion microbes during our lifetime. So we have more microbial cells than human cells. So it would actually be a part of our weight even that shows up as microbes. And these are, it turns out, critical for our own lives. So looking at just 4 major areas, our number of genes gets expanded from our 23,000. Just in your mouth alone, you have on the order of 4 million different genes, 1,000 different species. And so this new field of the microbiome is starting to expand what we know about the human body and human genetics. It turns out a number of factors contribute to disease. There's genetic susceptibility for infections, but it's also related to the quality of our immune system and all these microbes that we have associated with us, plus the rest of the environment. So to solve these diseases, we have to look at all of those. And here's just a list of some diseases associated with the microbiome and different microbes, everything from esophageal cancer to colon cancer to stomach cancer are all known to be related. But some of these other things with maternal health are showing up to be an important part of the microbiome. Just looking at esophageal cancer, it's the fastest growing malignancy in the US. It turns out that the individuals with the esophageal cancer have a unique set of microbes. It's not clear if they cause the cancer, it's just associated with the changes. But look at this change in esophageal cancer compared to the increase in the other tumors. So it's changing quite dramatically. And if the microbes are a key to understanding, also it could be a key to prevention and a cure. Now, it's not just a cause of disease. While you're eating this food, the microbes are in fact helping you digest it and creating new chemicals. And so actually looking at some of these microbes and the chemicals, so our 23,000 genes come up with maybe 300,000 different proteins. our bodies can make around 2,400 different chemical compounds. So if after this lunch we were to take a blood sample from each of you, we'd find around 500 chemicals in your bloodstream. 60% of them are from our own human gene metabolism. 30% are from this diversity of biology that you're consuming right now. But 10% are on the order of 50 chemicals circulating in your bloodstream are from bacterial metabolites. These are chemicals produced by bacteria. We have no idea what their role in our wellness, disease, how we feel. So this is a whole new area as we expand our understanding of humans. Now, if you look at these projections, and this is from a recent WHO publication, trying to look forward over the next 20 years, you can see the communicable diseases and maternal health issues, the deaths from these go down dramatically from 27% to about 14% over the next 20 years. But the diseases now that we're facing much more in the US become much more common in the world. Heart disease and cancer take up to become a huge percentage of the projected deaths in 20 years. So understanding our risk for disease and how to prevent it from genetics will play an important role in this. This is a slide, I'm sure it's probably impossible to read up there, but this is the 33 million people with HIV right now. Roughly 3 million new get added each year, and about 2 million die. And we look at the global map of these. Obviously, Southern Africa and the former Soviet Union are very high densities of HIV, creating challenges, obviously, to treat it. But the best way is to try and prevent it. And there's been a lot of work with vaccines, but none of them have been very effective, in part because the virus metabolite changes its genetic code so rapidly in response to our immune system and other factors. And there's a lot of diversity now of the viruses, so trying to make an antibody that works against one particular form of the virus has not been effective at all. In fact, there's lots of diseases like this. The common cold has never had an effective vaccine because the virus changes its genetic code so quickly. But we do have some new approaches now that might address some of these areas with diseases like malaria, HIV, and what we're starting with H-flu. And that's what BJ said. We just announced earlier this year the first synthetic life form. But this ability to write the genetic code, and what we did in this case is we wrote from scratch, from 4 bottles of chemicals, the complete genetic code of a bacteria, put that piece of DNA in like a new piece of software, and it converted a cell into the species defined by that DNA. But this ability to write DNA is now an important aspect. When we read the human genome 10 years ago, we were digitizing biology. So we took the 4-letter genetic code and converted it to ones and zeros in the computer. Now we can go the other way. We can start with these ones and zeros, write whole new genetic code, and come up with new approaches. This is actually a picture of the first synthetic cell that we reported, and it's gone through over 1 billion replications and could go on forever. We now can start with the computer and have robots write the genetic code, and we're trying to use these for a number of areas. We started 15 years ago with Chiron, and then they got purchased by Novartis to make the first genomic vaccine. It just finished phase 3 clinical trials in Europe for meningitis. Many of you know meningitis is one of those diseases. By the time you diagnose it, it's too late. So the best thing is prevention with a vaccine, and this new vaccine is looking extremely good. We're using now the new approaches to write the genetic code to see if we can make new flu vaccines at least as fast as the new flu strains appear. So my institute, along with others, is constantly sequencing the new viruses as they come out. We can, in 24 hours or less now, make new seed stocks. It took about 6 months to get the H1N1 vaccine out. We can lower that to a very short period of time, particularly if the FDA approves these new cell-based, getting away from egg-based production of vaccines. So these tools are going to get their first test in the next couple of years as a new, very rapid way to make vaccines. But because we can write the genetic code, we can make a lot of variants in a single cell and maybe cover the whole range of variation covered by HIV or rhinovirus or some of these other approaches. So we're going to be applying these in an exciting way. We can actually, at Synthetic Genomics, we have software for designing the software of life to try and create cells for new purposes. And part of health is having a healthy planet. And so we're also trying to use these to make new sources of clean energy and clean water. And last year, we announced our program. ExxonMobil's put up $600 million to help fund trying to use algae to produce fuel from carbon dioxide. But if you look at this table of what the different plants produce, there's almost a 500-fold range from what you can get from corn to right now what you can hypothetically get out of algae production. So we're trying to use these approaches to make totally new sources of food, fuels, chemicals, plastics, just starting with an energy source that we have abundance of on this planet, that's sunlight, and something we have in too much abundance right now, carbon dioxide. So we're trying to use the carbon dioxide as the basis of making new carbon-based chemicals. So we're taking a number of approaches, from the human genome to expanding understanding the human repertoire of genetics, to the microbiome, understanding how all these microbes play a role in our lives, both in health and disease, and then using these new approaches for new vaccines and hopefully some major treatments for the planet itself. Thank you very much.